Welcome to AP Biology. This video is on fermentation, anaerobic respiration, and other molecules that can enter cell respiration. Aerobic cellular respiration requires oxygen in order to produce ATP. Without oxygen, the electron transport chain cannot properly function. It needs oxygen as its final electron acceptor. Without oxygen, glycolysis is going to be coupled with either fermentation or anaerobic respiration in order to produce some ATP. Anaerobic respiration is essentially the same thing as aerobic respiration, except that it's going to use a different final electron acceptor. For instance, some bacteria use sulfate as their final electron acceptor. The process of fermentation is only going to use substrate level phosphorylation in order to make ATP. No oxidative phosphorylation can take place without the presence of oxygen. There are two types of fermentation: alcohol fermentation and lactic acid fermentation. The actual process of fermentation does not produce any ATP. Instead, the goal of fermentation is to allow glycolysis to happen over and over and over again, or to perpetuate glycolysis. Every time glycolysis takes place, 2 ATP are made during substrate level phosphorylation. In order to perpetuate glycolysis, what's going to happen in fermentation is that NADH is going to be converted back into NAD+. This is going to allow fermentation to take place again. We begin with alcohol fermentation. Alcohol fermentation takes place in yeast in the absence of oxygen. Yeast are still going to begin with the process of glycolysis no matter what. Glycolysis happens with or without oxygen. During glycolysis, glucose is broken down into 2-pyruvate. This produces 2 ATP, and this also produces 2 NADH. During the process of alcohol fermentation, the pyruvate is going to be converted into a molecule called ethanol, which is alcohol. You can see that pyruvate is a 3-carbon molecule, ethanol is a 2-carbon molecule, so the other carbon is going to be released as carbon dioxide. When fermentation takes place, the NADH is going to be converted back into NAD+. That's the real goal of glycolysis. That's the real goal of fermentation. The goal of fermentation is not to produce carbon dioxide and not to produce ethanol. It's to take the NADH and convert it back into NAD+. Once that NAD+ has been regenerated, glycolysis can occur again. We use yeast in order to produce alcohol and in order to allow bread to rise. You can see why both take place. Bread rises as a result of the carbon dioxide that is released. And alcohol is created using the end product, ethanol. Ethanol is essentially just drinking alcohol. The second type of fermentation is called lactic acid fermentation. This takes place in some bacteria. We use this bacteria to make things like yogurt. And lactic acid fermentation also takes place in muscle cells. Again, it begins with glycolysis, making 2 ATP, making 2 NADH. Glycolysis is going to cut glucose in half. It's going to make 2-pyruvate. In this case, during lactic acid fermentation, pyruvate is converted into lactate or lactic acid. Both are 3-carbon molecules, so no carbon dioxide is going to be released. This lactic acid is what makes our muscles burn during exercise. Again, this fermentation has the same goal, to take the NADH and convert it back into NAD+, to allow glycolysis to occur over and over again, making 2 ATP per clip. If we compare aerobic respiration with fermentation, aerobic respiration makes far more ATP per glucose, 32 ATP per glucose. And if you remember, that 32 is going to be made primarily using oxidative phosphorylation. This takes place during the electron transport chain and chemiosmosis. The process of fermentation is only going to produce 2 ATP per glucose, but can be beneficial if the organism is not in the presence of oxygen. There are different types of organisms. Obligate anaerobes. These are organisms that carry out fermentation or anaerobic respiration and cannot survive in the presence of oxygen. So aerobic respiration is not an option for these organisms. Yeast and many bacteria, and really our cells, can be considered facultative anaerobes, meaning that they could survive using either fermentation or aerobic cellular respiration. For a facultative anaerobe, like our cells, pyruvate really represents a fork in the road. In the presence of oxygen, pyruvate is going to enter the mitochondria, where aerobic cellular respiration will take place. Without oxygen, pyruvate will remain in the cytoplasm and will enter the process of fermentation. Evolutionarily, glycolysis is a really important process. It's the first process ever used to make ATP, to make energy. Life evolved 3.6 billion years ago. Oxygen was only available in the atmosphere about 2.7 billion years ago. So during that period of time, from 3.6 billion years ago to 2.7 billion years ago, these simple prokaryotic organisms used glycolysis in order to produce ATP. So glycolysis is an extremely ancient process, and the first process used to make energy. We always talk about glucose going through the process of cell respiration, but glucose and carbohydrates are not the only types of molecules that can be broken down to make ATP. Proteins can enter cell respiration, so can fats. In order for proteins to enter cell respiration, they first need to be broken down into their amino acids. Once they are broken down into amino acids, the amino group needs to be removed. The amino group is the NH3. Nitrogen cannot enter cell respiration. The process of removing the amino group is called deamination. Once this amino group has been removed, the resulting molecule could enter cell respiration at various stages. Fats can also enter cell respiration. If you remember, a triglyceride is made up of a glycerol and 3 fatty acids. That glycerol can be used in glycolysis, and fatty acids are broken down through a process called beta oxidation. These long chains of hydrocarbons that we call fatty acids are broken down into 2 carbon molecules through the process of beta oxidation. These 2 carbon molecules become acetyl-CoA and can then enter the citric acid cycle. You can see that 1 glucose is only going to make 2 acetyl-CoA, but a triglyceride can make many acetyl-CoA. So an oxidized gram of fat is going to produce more than twice as much ATP as an oxidized gram of carbohydrate. That's why there's so many calories per gram of fat than there is in a carbohydrate. Here you see both of these processes taking place. You see that the proteins are broken down into amino acids. The amino group is removed through deamination. The resulting molecule can enter cell respiration at various points. Fats can be broken down into glycerol and the fatty acids. Glycerol can enter glycolysis, and the acetyl-CoAs made during beta oxidation can all enter the Krebs cycle or the citric acid cycle as acetyl-CoA. The products of each stage of cell respiration can also leave cell respiration and enter anabolic pathways. These anabolic pathways build the important molecules of our body, build the parts of our body essentially. This process in which the parts of our body or these large molecules are built is called biosynthesis. Finally, since cell respiration is a metabolic pathway, right, it's a catabolic pathway, it needs to be very closely regulated. Regulation is performed through feedback inhibition. If you remember, feedback inhibition is when the end product inhibits the first step in the metabolic pathway. The ultimate end product of cell respiration is going to be ATP. So when ATP builds up in the cell, we no longer need cell respiration to take place. ATP is going to work as an inhibitor. In this case, it's going to inhibit the first enzyme in the metabolic pathway. This enzyme is called phosphofructokinase. It's an example of an allosteric enzyme, an enzyme that can be turned off through ATP. If AMP builds up, that's only going to occur if you have an extreme lack of energy. AMP is going to stimulate phosphofructokinase. It's going to turn it on. So ATP can turn it off, AMP can turn it on, and the cell uses this to very tightly regulate its concentration of ATP. This concludes our video.